Hi. Hello. Thank you so much for the opportunity to present our work. DNA in our cells is continuously damaged on a massive scale due to numerous exogenous and endogenous genotoxins. It is estimated that around 100,000 DNA modifications occur per mammalian cell on a daily basis, including DNA breaks, DNA oxidation. DNA modifications provide precursors for mutations, therefore driving carcinogenesis. Simultaneously, many anti-cancer drugs induce DNA breaks to kill rapidly dividing tumor cells. It is becoming clear as well that DNA modifications beyond cytosine methylation and its derivatives epigenetically control gene expression. DNA damage is also involved in neurodegeneration and aging. In fact, DNA modifications and DNA damage signaling is causally linked to probably all phenotypic manifestations of aging, including dysfunctional telomeres, dysfunctional mitochondria, senescence, and inflammation. And many premature aging syndromes are caused by defects in DNA repair. This evidence and a lot of other evidence suggests that DNA damage is an important factor in aging and probably the driver of aging. However, due to technical limitations, there is a gap that we do not have age-resolved genome-wide measurements of DNA modifications and single nucleotide resolution. And we developed methods to reveal which, where, and how much DNA damage is in the genome, and this will help us understand molecular mechanisms behind biological impacts of DNA modifications, including in aging. We also want to establish genome-wide DNA modification measurements for toxicology testing and precision medicine. We also want to build an aging clock that would be implicated in the very core of this process, in DNA damage. So today I will present our two recent methods, ABasic for mapping chemotherapeutics-derived DNA breaks, and ClickCodeSeq for sequencing oxidized guanines and abasic sites. And at the end of my presentation, I will share our initial data on DNA oxidation in aging. In the first study, we wanted to reveal molecular mechanism of the natural product and approved anti-cancer drug trabectedin. We used comet chip assays and gene knockouts, and we figured out that trabectedin DNA adducts specifically derail transcription-coupled nucleotide excision repair, or shortly TCNER, by inhibiting the second essential endonuclease called XPG. So this repair pathway, TCNER, is crucial in aging because deficiencies in it cause premature aging called cocaine syndrome. And based on this discovered mechanism, we devised a tool to profile TCNER activity genome-wide by actually mapping these breaks induced by the drug trabectedin. Our method includes isolation of DNA fragments adjacent to the breaks with an adapter that also has biotin. Then we index these DNA fragments to account for cellular distribution, and we sequence these fragments locating the original positions of the DNA breaks. With this method, we discovered a massive break formation when we treat wild-type cells with trabectedin, and these cells have TCNER functional. In the model of cocaine syndrome, in the CSB knockout, we virtually do not see induced DNA breaks. Most of the trabectedin-induced breaks occur on the transcribed strands of genes, so in these green boxes, and as a function of gene expression. And DNA breaks peak at the beginning of the gene bodies. And there is an interesting swap right upstream of the transcription start site. Breaks occur on the non-transcribed strand from the gene body perspective. And this may be evidence that TCNER works beyond genes and may be connected to so-called divergent transcription from promoters. We also generated evidence that the essential endonuclease of nucleotide excision repair, XPF, has a sequence preference. So we observed a G enrichment downstream of the induced breaks, which is something new. So as a summary of this study, we found out that trabectedin induces DNA breaks in highly expressed genes in a dependency from TCNER. And this mechanism provides a basis for profiling TCNER activity genome-wide in aging. However, only in model systems, not directly in humans, because this drug is rather toxic. But what is more promising as an aging biomarker is endogenous DNA damage. including DNA oxidation. We can map 8-oxoguanine, one of the most common forms of DNA oxidation, with a method called ClickCode-seq. In this method, we recognize 8-oxoguanine enzymatically, and we cleave it, creating a gap. We fill the gap with an alkyne modified nucleotide and click to this alkyne an oligo that has an azide and a biotin, so that we can enrich the library, sequence it, and identify the genomic locations of 8-oxo-G. And it is especially pleasant for me to mention click chemistry in the whole of this university, because this university's professor, Morten Meldal, received Nobel Prize for click chemistry in 2022. So we also supplement our method ClickCode-seq with fluorescence-based quantification, and this is very useful for library preparation process. To increase the specificity of our method for 8-oxo-G, we block other abundant forms of DNA modifications, including abasic sites and breaks. And if we need to use this method to map abasic sites, we use different enzymatic treatment and we block the breaks. So using ClickCode-seq, we discovered that the propensity of guanine to be or to remain oxidized is the smallest at CpG dimers compared to other dinucleotides, and this may reflect a relationship between guanine oxidation and cytosine methylation. When we look at the trinucleotide context of guanine oxidation, we identified that the signature of guanine oxidation, these orange bars, they are very similar to the lollipops, which is the cancer mutational signature 18, which is known to be associated with reactive oxygen species. Actually, compared to all mutational signatures documented in the COSMIC database, signature 18 is the closest match to the 3 replicates of our guanine oxidation mapping. So this is an expected result and confirmation that what we mapped makes sense. And matching DNA damage, DNA modifications, and mutations seems to be promising for discovering the etiology of cancer. So we surprisingly identified that the abasic sites induced by the anti-cancer drug irofulven have a spectrum across trinucleotides, which is very similar to the cancer mutational signature 40B. 40B was just recently reported, and it is associated with kidney cancer, and it was found around the world. It is not yet known what is the cause of this mutational signature. It's like seeing smoking-induced mutations, but not knowing that smoking causes cancer. And according to our observation, a chemical or a metabolite, which is similar to irofulven, it may cause this mutational signature 40B. Another interesting observation that we made with ClickCode-seq is that with increasing gene expression, the guanine oxidation increases on the non-transcribed strand compared to the transcribed strand of genes. And this is seen also here, only throughout the gene body, and evaluated here statistically. And a reason for this may be the fact that the non-transcribed strand during transcription is exposed as a single strand more than the transcribed strand, which is in the R-loop structure. An additional idea may be that the 8-oxoguanine is repaired in a transcription-coupled way. And indeed, when we mapped abasic sites induced by a known TCNER-specific drug irofulven, we also see a very similar strand bias. So this indicates that the guanine oxidation may also be, or our data may indicate that 8-oxo-G is repaired by TCNER, which is an emerging view in the DNA repair field. The ultimate place, I think, to look for DNA damage, DNA modifications, is the mitochondrial genome because of the proximity to the reactive oxygen species. We found that the minus or heavy strand of the mitochondrial DNA accumulates more guanine oxidation and also abasic sites that are derived from adenosines. And this is in line with the currently dominant view on the replication mechanism of mitochondrial DNA. So in this model, strand displacement model, the minus or heavy strand is exposed as a single strand for a longer period of time. Therefore, it may be more vulnerable. So I have shown to you that the DNA modifications that we map with ClickCode-seq correlate with functional genomic features. in the genome. Therefore, what we map is likely not noise. And motivated by this, we embarked on profiling DNA oxidation, guanine oxidation, genome-wide in human aging using human blood, with the ultimate goal of discovering a novel DNA damage-based biomarker of aging. We have profiled so far 132 blood DNA samples. We have 20 or 30 remaining samples left, and we already see promising correlations between the oxidation of individual genes in age. In a linear combination of different genes, we expect to build a good clock. As a summary, we are close to building this new clock of DNA oxidation using the healthy human data. We are currently also profiling DNA oxidation and DNA breaks in mice using healthy animals, also calorie-restricted ones, and with premature aging. And my ultimate desire for future work is to link genomic patterns of DNA oxidation and DNA breaks with physiological parameters, morbidity and mortality. I'm more than happy to be, and lucky to be working with this fantastic team at ETH Zurich and Center for Genomic Integrity in UNIST, Republic of Korea, with Professor Shanna Sterle and Orlando Scherer. And please come to the poster of Navneet Singh. She will talk about photoaging. I'm also very grateful to the funding sources, Swiss National Science Foundation and Longevity Impetus Grants, Evolution Foundation and Rosenkranz Foundation. And thank you so much for listening.
I have a question. Yes, perfect. So I was wondering if you looked at predicted secondary structure, if that's prone like G-rich sequences, G4 quadruplexes, or other types of secondary structures that you find in DNA.
Yeah, that is indeed very interesting. And there was a recent paper where authors observed that ATOXG is accumulated in G quadruplexes. We are looking into this, but we should be very careful because it's super artifact prone. The way library prep is made and the secondary structure may be affected during the library prep. So there should be a range of controls for that.
But you can do it by predicted sequences, right? So,
yeah.
Okay, very exciting.
Yeah, thanks.
Thank you for your data on mitochondrial DNA oxidation. I was just curious whether the mitochondrial DNA oxidation clock might be more sensitive to age-related changes than the nuclear DNA, since it doesn't have good repair mechanisms. Can you comment on that?
Yeah, thank you for this idea. Indeed, I suspect that it may be the case, and we are definitely looking into it. So we will look into this trend asymmetry changing through aging, and it may be connected to the last talk of the previous session, where in rodents it was observed that the expression of genes changes with aging. And the data that we have, that the majority of DNA modifications occur on the heavy strand of mitochondrial DNA may be related to that, because the heavy strand is the one on which RNA polymerase is moving for the majority of genes. And so so RNA polymerase may stall on ATOXG, or maybe on some repair products of ATOXG, and may decrease gene expression. This is something that we are very interested in. Thanks.
Thank you so much for your wonderful talk. Help me thank you, Vakil, for his talk.